I'm going to go ahead and turn it over to David and Cyril um, for their presentation today. And again, just a reminder, please stay on mute and I'll be monitoring the chat. Thank you, guys.
Hi, everyone. Thank you, Brittany. My name is David. I'm logging in from the state of Maine. My colleague Cyril, who you'll meet in a minute, is sitting in the country of Belgium. Um, really happy to be here. Thank you for having us. Happy to share both for purposes of publicizing what we do and getting feedback, because as you'll hear, what we're doing is pretty new and pretty different. And we started in the United States just over 3 years ago. As you'll hear, our science team developed this methodology and the tools we use in Europe originally, almost a decade ago. Quick introduction. I am trained as an environmental policy analyst and environmental economist, and I lead a number of our biomonitoring projects, we call them, around the US. Cyril, you want to give a quick intro to who you are?
Yeah, good morning. So my name is Cyril. I am, so as David said, talking from Belgium, where the headquarters of biodiversity are. I am an agronomist. I studied tropical agriculture, and I've been working for biodiversity for 5 years now, trying to develop the projects all around the world. We are so far in 22 countries, and with David in several places in the US where will be, that David will present you a bit further. I'll let David introduce a bit what we're going to talk about.
Awesome, thank you. I think we're going to talk for a little over 30 minutes at the start here, give an overview of a number of things, including sort of what we focus on. I'll introduce a little bit of our US work, including some coastal zone work. Cyril will give the bigger picture, and then we'll dive into a couple more case studies in the United States, specifically, amongst others, a lot of work we're doing along the Klamath River region in Oregon we've chosen to focus on today, related to biodiversity restoration and the removal of the dams and sort of what's going on the river out there, restoration of salmon habitat, tracking of agricultural runoff, as well as native and invasive species impact along the coastal lands and with the local communities, including some Native American tribes and others. So we're gonna tell that story towards the end, but just a hint at sort of the stuff we're going at. I guess the general topics are biodiversity monitoring and reporting and verification on the ground. With partners and clients, we only work in support of local stakeholders. So while we provide a centralized set of services and analytics and tools and data, it's all intended to serve whoever's on the front lines of the local action, and can use this information for real biodiversity impact and human health impact. So we'll talk about that. We'll talk about data on plant species, data on contaminants, how that data can roll up into baseline information and risk factors and habitat classifications, regional impact classifications, action recommendations, tracking metrics over time, those sorts of things. I guess the final piece of my introduction then, and I'll turn it over to Cyril, is that what you're going to hear is that our primary source of data is samples collected by bees, tens of thousands, hundreds of thousands of bees across the landscape, aggregating to tens of millions of samples per month on sites of 1,800 acres, more or less, on average. I guess the final piece in terms, again, the US context is that we decided to begin to introduce our methodology in the United States about 4 years ago. We did an initial test case with the land trust in Maine along the Kennebec River. And it then started developing what we call full project implementations in Maine with Maine Audubon, with USDA's Natural Resource Conservation Service through a conservation innovation grant at sites in Montana, Minnesota, and Oregon, along the Klamath rivers I discussed, on a solar farm in Texas, through some industrial restoration work at a Superfund site, and some other cases that we'll discuss. So Cyril, if you could please give the big picture, and then I'll zoom back in on these US cases.
Thanks, David. 10 years ago, by two Belgian, one is a scientist, the other one is an entrepreneur, and they joined forces because they believe that it's possible to create value with biodiversity, create value for public entities, for corporates, and what we actually do is trying to regenerate biodiversity on sites, in municipalities, on company sites, and always with a view of how can it create value, how can it add value, and not be considered as a cost. Because as you know, as restoration and protection professionals, biodiversity and nature is often considered as a cost. And so the idea was to change that mentality, change that point of view, and really show that with combining expertise, combining nature-based solution and some data, and now in the last years, adding some AI and machine learning technology to help it go faster. And by the involvement of stakeholders, it was possible to create value with biodiversity. It started 10 years ago. Today, we are working in 22 countries. We are covering with our tool that David mentioned quickly, and I will go a bit deeper into how it works. About 320,000 acres. We're a team of 30 people plus 60 to 70 local beekeepers. We have about 100 active projects in the US, but also in mostly in Europe, but also in Africa and in South America. And we are the largest employers in the world with our 30 million employees, as I call them. So what we do is that we help public entities and companies either on the strategic level all the way to the operational on-site projects to assess, advise, and act in terms of biodiversity. I will explain way more detailed what we do. But so we have this strategic view. Some of you might know the business model canvas. It's a big canvas on which you try to understand how develop a business model. Well, we adapted it to the biodiversity canvas so that we follow a path of several steps, and in the end we see on which ways, on which areas we can create value with biodiversity. The on-site projects is the one that I will present more in detail, is how to assess, because we're very convinced that if you can't measure something, it's impossible to improve it. And so what we do is that we created several tools. One of them is our flagship tool, I would say, to really monitor and assess the state of biodiversity, but also the state of pollutants use. I call it the Google Earth view. So when you go on Google Earth and you type an address, you first see the globe, and then you deep dive into the place that you typed in, that you're looking for. And so what we do is that we start with a SaaS platform, which is remote sensing, and will do a multi-site analysis of the risks and opportunities in terms of biodiversity, but also the risks of finding some kind of pesticides in an area that you can define yourself. It will, by example, help to understand what are the pressure of some industries or some residential areas on some protected areas. What are the industries that could affect your the protected area in which you're working? So this is a tool that uses cartographic analysis, but also a lot of our data, the data that we've been collecting in the last 10 years, and that's combining all that to really give you a prioritization of the sites on which you might have an issue, or on which an industrial company, for example, could have an impact. And then you go down to the territorial level, where we use, I heard that 2 days ago, is the BAS, so the bees as a service. So we collaborate with honeybees to gather samples from the environment. This is the biomonitoring. So we cover 1,800 acres on which we can measure plant diversity, but also a large amount of pollutant. And then when you arrive at the address that you've been looking for on Google Earth, you are at the parcel level, at the site level, at a very, very localized point where you can, where we can work either with solitary bees, with the bee hotel, or with eDNA, underground, above ground. And when you have a view of satellite view, territorial view, and parcel level, above ground and underground, then you have a global understanding of what are the risks, what is good, what is not good in terms of plant diversity, in terms of biodiversity, ecosystem health, and in terms of pollution. And so then we can advise, deliver KPIs, and follow up the evolution during the time. I'm going to talk about the biomonitoring. Biomonitoring is a unique tool that was created by the founder of one of the co-founder of BeeOdiversity, which is the Dr. Pak Kim Nguyen, who is an agronomist specialized in biodiversity and pollination. And he found out during his PhD thesis that bees can actually be, we can collaborate with bees to collect samples from the environment. So bees will forage on a radius of about a mile around the hive, which covers, it's 700 hectares, it means 1,800 acres. And they will visit about 4 billion flowers each year. I'm talking about years here, the bee year in Western Europe, which probably is the case in the northern or in the central states of the US, probably not in the south, is between April and October. Of course, it will be a bit longer in the south, a bit shorter when you are in maybe Dakota or Wyoming, where the winter is a bit more important. But the bees, 50,000 bees on average in a hive, will visit several billion flowers per year. And from those flowers, they will bring back pollen and nectar to the hive. The nectar they'll transform into honey. The pollen is the source of protein. What we did is that we created a small device that we place at the entry of the hive to collect pollen on a daily basis. So we have virtually access to 4 million points on 1,800 acres. And by analyzing the pollen, as you know better than I do, pollen is outside the plants, so it can capture the atmospheric pollutants, but it also contains the DNA of the plants. And so by analyzing the pollen, we can measure more than 500 pesticides, we can measure nitrates, phosphates, but we can also measure heavy metals, pH, dioxins, PFAS that are present in the atmosphere, in the air. And through the DNA analysis of the pollen, we can measure the plant diversity. And by crossing all the results and using a split bee year, I call it the bee year, as I said, between April and October, we have 4 periods of 6 weeks. We can first, by crossing the data, we can understand what's the origin of a pollutant. But we can also identify periods where there's a lack of plant diversity. And when there's no flowering plants, there's no insects, and there's less birds. And so the whole ecosystem is kind of at risk. And so by understanding when there is a lack or shortage of plants, or when there's less flowering plants, then we can kind of rebalance the food supply chain, the food supply curve for pollinators, which flowering plants are one of the first part of the chain. And so this is what we do. All those data is processed by some technological programs and algorithm. I don't understand about it, so if you have more questions, I'll be happy to ask my colleagues and answer those. And you will see that using our data, you can even go down to understanding what kind of birds could be hosted by your reserve. And David will talk about that question that we had in Maine. Because of the plants that are in the area. And you will see that it had other, that was the main focus, the first focus, but it had a huge other consequence when the bees started to gather data on that place. What's very interesting is that because we have about 150 sites today, we can have a benchmark of your site. So we can put your sites on a scale between all the sites that we measure, or all the natural sites that we measure, or all the sites in the US that we measure. We can see where it stays, where it stands compared to the maximum size when we talk about the number of species, or the minimum size when we talk about the number of pesticides. And you can follow this during the year. And it's a very interesting, you have like, for the plant diversity, we have 7 indicators that you can follow, and you can follow them throughout the years. And so you can see the impact of your actions. You can see the impact of, by example, a new residential development not far from your reserve. You can see a lot of things that can that are really interesting to understand at a landscape level, 1,800 hectares, 1,800 acres, sorry. What is happening on the site. We not only send you an Excel sheet with the results, we interpret them, and then we provide advice and actions. You can also just ask for the raw data and then, of course, interpret it yourself, because you know the site much better than we do. This tool was awarded several prizes because it's there's no other tool that can simultaneously collect so many data on such large surfaces, and that enable us to analyze so many elements. You could have like sensors all over the place, but they only analyze 1 or 2 elements. This is why the biomonitoring is used by a very large array of public and private entities around the world. I was speaking about the parcel level monitoring. We developed here what we call the Bee Hotel, which is a solitary bee hotel scientifically designed to host Megachilidae taxas of bees. And depending on the hole that is filled and the way it is filled, we can determine which bees are in the ecosystem. It works on a much smaller surface of about 100 acre, 40 hectares. It's what 350 meter is about 300 yard, I would say, a bit more than 200 yard around the bee hotel. So it's a much smaller scale. Of course, solitary bees, as you see, we can open the box and we can take the tubes and we could take the pollen and analyze it. Of course, this is destructive, so that's something that you might or might not like, but I just wanted to mention that it is also possible to collect pollen by solitary bees, but of course, solitary bees are much more selective in their diet, and so it will not give the same results as the biomonitoring. We are launching now a project where we combine this biotel with soil samples, with plant sample analysis through DNA. The pollen sample, as I just mentioned, we have added some sensors on the biotel. We are going to deploy this on 200 sites, and the goal is to launch an AI-powered predictive platform that will enable us to create an app, and that this app will, just by taking a picture of the face or just by going in front of the faces, will give you a score of the health of the ecosystem. We do believe that solitary bees and the Megachilidae in particular are a very good bioindicator, and that we can use them just by knowing who is there, who has laid an egg in this bee hotel, what is the state of the ecosystem. And I mentioned soil, plant, and fauna eDNA, so the extraction of DNA parts of DNA from soil samples, from plants, from other matrices, help us to understand what is the state of biodiversity, what microbes are in the soil, and what function do these microbes provide to the soil. For example, when we're talking about agriculture. I just wanted to explain you one or two small stories about what we have done. We are here in a small coastal city in Belgium. So this is the North Sea. You're, I don't know, 20 miles from the UK here. And we have been monitoring on 5 sites for the last 10 years. So we have a lot of data in that municipality. And the first year we came, we found very high concentrations of lead on those 2 beehives that are in red now. Excuse me. I'll explain how we managed to do that. But we looked at, we tried to understand. We did a small, like, Sherlock Holmes investigation on why would there be lead on those 2 hives and not on the other, and that's the power of the tool. Well, we found out that some ships couldn't connect to the electricity supply system from the port, from this industrial port that is just here. And so by letting the engine run, they were burning heavy fuel, and so the lead was coming in the hinterland. We found some pesticides, some linked to golf courses, some linked to fruit, apple, and pear crops. And so we organized. We've been organizing now workshops with farmers for the last 5 years to decrease the amount of pesticide that they are using. We did it first with apple and pear producers. Now we've been working with potato producers, and the next step is to work with corn producers because there's a fungicide that they're using at very high level in the last years, and we're trying to understand how they can change that. And then we found that there was a lack of plant diversity. And so we talked with the municipality, and we decided to involve the local people. And so we're distributing about 1,000 seed bags that people are planting in their garden. Very simple solution. But we managed to multiply by 4 the amount of plant species that we found in 3 years. And you see that with this tool, what's interesting is that you can show the impact of your actions, but you can also detect an issue. And just right after the first period of 2017, we found out that there was a huge decrease of plant species, of the number of plant species. So we went back to our data and we talked with the municipality, and we understood that actually the way the green zones management services from the municipality was working was not right. And so we trained them, and now we never have this problem anymore. And so we now stay at about between 45 and 50 plant species all year round. And so we have a very, very nice ecosystem where we rediscovered some bee species, some wild bee species that hadn't been seen in Belgium in 10 or 15 years, for example. So you see that we changed a lot of things, and this is really because we help the politics to make decisions with scientific metrics and scientific data. I'm just showing this. This is an example of pollutants monitoring. It shows the periods. Yes, it starts by P3 because on this particular project, we were working from P3 and the third and fourth period of 2021, I think, and the first and second period of 2022. Sorry, it's a bit a mess, but so you see that we can really have we have very simple visualization that says it's good, it's not good, and then we go into the detail. Why is it good? Why is it not good? Is it because of the amount of metals that we found, the different amount of heavy metals? Is it because of the concentration of the heavy metals? And so we will go deeper and deeper in the analysis, depending on the data that we receive. I want to speak now about what we do in terms of biodiversity restoration. We had a project. This is a project in South Africa, as you can see, where a large brewer is growing hops in a very specific ecosystem. This ecosystem is harmed by eucalyptus, by invasive species that are pumping a lot of water, which makes the hop harvesting more complicated. So what we decided to do is we measured on 5 sites the plant diversities. We looked at no, sorry, this is another site. Excuse me. This is a site in Paris. Very interesting data. You see that we have 5 sites in Paris from a private company, and they are looking at plant diversity and the evolution of plant diversity. And we saw that 2021 was a very wet, very cold summer in Europe, in Western Europe at least, and in the north of the Loire River. And then 2022 was very dry. And so we have a real decrease of amount of plant species. And then we see that 2023, we have a huge increase compared to the other sites of plant species. And what happened actually is that there's the Olympics. You might have heard of it. It's starting. It started 2 days ago. Everybody is looking at it, and so Paris started planting a lot of flowers. And see what we see directly. We go from 18 to 44, 38 plant species. So you will see the influence of actions directly on those data. I will go back to what's interesting also is that we will look, at the proportion in wild species, because it's very important to have a lot of plant species, but if they are only ornamental, or if they are only not wild indigenous species, if they are invasive, for example, or if they are ornamental, then they are not interesting for wild pollinators. They're not interesting for wild and indigenous animals, and for the local ecosystem. And so this is one of the indicators that we follow in terms of plant diversity. This is I'm going back to South Africa. Sorry, there's a small mix in the slides. What we see here is that we had a site that is completely natural with almost no invasive species. We have sites where there's a lot of invasive species, and the sites where the brewer worked with WWF to restore the zone. And we could show that the restored zone had more plant species than the others. And so that the actions that were taken were performant, that they were useful. And this is something that you might look for, is some data on what I am doing some actions. I see that it works, but what are the scientific data behind it? This is a tool. This is what the biomonitoring can help you do that. we're looking at plant diversity, but we combined it also with soil health. And so what we did is we compared here 3 different places, as I said, the natural place, the invasive place, and the hubs and restored place. And so you could decide to have a place that is very natural as a 100%, and then you could go in the city next door where there's only very intensive agricultural, and say this is my 0%. And then the places where I'm working, I'm going to try to put them on a cursor and see where I stand and see how I can go as close as possible to the natural place. What might be interesting also is that you can measure the contribution of a site on the territory, because if you have site audits and then you compare it with the species of the biomonitoring, then you can compare or calculate the contribution of one site on the territory. This is here another example, another indicator that we give is what are the dominant species? Are there several dominant species, meaning there are more than 5% of the pollen that comes from those species? If you have only 1 or 2 dominant species, then it's a risky ecosystem, because if that dominant species dies for whatever reason or is harvested, then there's no food anymore for the bees. And then, of course, we're looking at invasive or non-native species and which one are dangerous, and we try to help our clients to fight against it. I have a last idea, a last calculation. As I said, we work on the wild plants and the contribution of the plants. What I explained to you is that by knowing when you have an issue or when you have a lack or a shortage of plant diversity, well, it's much easier to know what to do on your site. Do you want to add more plants? No, you are in a reserve, so you don't add more plants. Do you want to try to change the management system that you have on the buffer around the reserve to improve those periods and have more of those plants flowering at the periods where you have a shortage? That might be a way to do it. And so we can help you understand how to change that. What is the contribution of a smaller site on a very larger territory? And really then also use this to communicate with stakeholders and involve more stakeholders in the protection of the biodiversity. I will go very quickly. We did I asked my colleague this morning to do a small exercise, and he gave me some example of identified genes by the biomonitoring that are present in some of the U.S. National Estuarine Research Reserves. So you can see a list here. We can come back to that. It's just to give you an idea that, of course, there's not a lot of bees in salty areas, or less bees in salty areas. And if you are working on coastal preservation, coastal zones preservation, then of course there's a lot of salty areas. But we still find some of the plants present in those National Estuarine Research Reserves, and it's very important to work on the edges. But David will explain that much better than I do. In the global ecosystem preservation. I will quickly go through to show you some example of heavy metals and pesticides diminution. What we did is really to communicate the results. So once you have results and you can explain where they come from, then you can go to those stakeholders that are responsible to the origin of the plants, of the pollutant. And once you have that, then by just making people conscious, you normally have very good results in terms of diminution of pollutants. I'm talking here about copper. Sorry, it's in French. Copper, of course, was applied to control disease. And by just explaining that we found copper above the maximum residue limit, well, the year after, it was a huge change, as you can see. So it's not bad to use copper, or I'm not for pesticides or anything. Don't take me wrong, but if you use it on a reasoned manner, it's at least better than use it at a very high level, for sure. So that's one. We'd rather have none of those pesticides, but when you have them, then at least it's good if we can keep them under the maximum residue limit, and if we can, or like this one, who was forbidden by the EU Commission, that we can stop it just by mentioning to local communities that it's still there and it's dangerous. It's been forbidden by the EU Commission because it's dangerous for the health of the one who applied it, for the health of your children, for the health of the birds, of any animals around it, and so you have to stop that, and that works well. This is I was talking about the captain. I was talking about the fruit producers in the small city earlier. Well, we worked specifically on Captain, which is a fungicide to fight the apple scab. And by doing 2 workshops with a farmer, you can see the very large differences that we made. Knowing that 2021 was a very wet year where they normally would use a lot of fungicides, I think that those results are pretty interesting, and that it could really be shown as a very good example on what, just by knowing and just by informing, you can already perform in terms of pesticides or of change in protection of the ecosystem. I will now let David talk about US cases, and then we will discuss or answer the questions you might have. I hope I've been clear enough. Sorry, my English is not my main language. Thank you.
Ooh, thank you, Cyril. That was a lot of information developed over many years. So thank you for the overview. Let me back up on a couple points for everyone. I'll probably go about 10 more minutes, and then we'll just stop and discuss, ask questions, whatever. So I guess a couple of big picture takeaways. What we would suggest, one is, actually, Cyril, why don't you go back one slide if you can? Yeah, just leave it there for a minute. Take everything we just discussed and back it up to say, okay, once you have the core data, which is really the point, there's a lot you can do with it. And so that's the direction that biodiversity has gone over the last several years. The core data, again, is if you can collect tens of millions of samples across the landscape and identify with a pretty solid degree of precision the plant species identification and diversity, categorize that data and the concentrations of these different contaminants or potential contaminants in the landscape, then what can you do with that information? And you've just seen Cyril illustrate many directions we've gone. In each of those cases, we're basically following the interests of the partners or the clients that we're working with. Let me say again, as background, I guess, of the different approaches and technologies that biodiversity has been developing in the United States, most of what we've done is what you call the biomonitoring, because our laboratory is Europe. So in the US, most of what we've done until now, which again is over the past 3 years, is the working with domestic honeybees that send out 20, 30, 40, 50,000 drones per day. And using the technology we developed to take daily samples to then analyze that for all the data we're discussing. The other one Cyril mentioned that I would highlight is what we call the Bee Hotel, which is the new approach we've just prototyped in Europe for wild and solitary bees. So a different technology, a different analytic approach, but looking at wild and solitary bees in the landscape, both to identify the health of the habitat, the presence of different species, the health of the habitat itself for those bee species, if it is in fact the pollinators that you're interested in. Some of the people we work with are partnering with the pollinators, but they have a different goal. Some is pollinator habitat itself, and to correlate the data they collect with the other data we can gather. And all of that is then matched up with your local information, right? It's local intelligence that makes any of this really relevant. One thing I want to mention finally here that Cyril didn't mention, but it's important as we consider this is we want everybody to understand there's a couple pieces of proprietary information in what we do. So in all of our work, the data belongs to the partner or to the client, 100%. There's a couple of the methodologies, however, where the intellectual property belongs to biodiversity. That has to do with the technique for the daily sampling, ensuring daily sampling for statistical relevance, rather than just sort of points in time, and some of the reporting and analytic methodologies. So we could discuss that more. Let me mention a couple of U.S. cases and then really focus on this Klamath River one for a minute and then just stop. The other cases, Cyril mentioned Audubon in Maine. What we did with Audubon was based on their interests, experimented with correlation of the identification of native plant species in some wildlands with the habitat that native insects need, as that connects with Audubon science around wild and migratory birds. So if, and probably you all know more than I, I'm looking at Tim, that the wild and migratory bird species prefer native insects, which need to produce in native local habitat. I just got distracted because Cyril put up the picture here of our colleague Tim Forrester. We wanted you to know that. Cyril, you're moving your mouse around and it's distracting me. So hold on. Sorry, everybody. Just hold there until I tell you to change, okay? Tim, who you saw, is a professional wetland scientist who's a member of our team in Maine. So in Maine, the Audubon piece had to do with correlating the native plant species with the insect habitat that correlates with wild birds. Pretty interesting. One thing that we identified there also was a plant species which is invasive to Maine, which had not been registered in the public databases as yet. So the bees actually went out and found a plant species that was not yet in the state registrar. So kind of interesting. Let's talk about Klamath because that's the slide that's up here. So this is sort of our biggest case study in the US now. We have, for 3 years, we now have 12 biomonitoring sites up and running around the upper Klamath Basin. The sites, I'm going to just mention a few characteristics, but basically we started because of philanthropy was interested in supporting some of the biodiversity monitoring work that has to do with the sensitivity of the region, the combined public and private government and philanthropic efforts to restore biodiversity in the region, and the removal of the big dams on the Klamath River and the restoration efforts around that. What we have going on now is within the range that you see on this map, which covers about 25 miles linearly in either direction. We have 12 full monitoring sites and 5 what we call discovery sites. The full monitoring sites are ones in which we collect the 4 periods of data each summer and do a comprehensive analysis. The discovery sites are ones that we've just set up this summer that are not doing the comprehensive daily sampling and broken into time series across periods, but just collecting a large volume of pollen at one site to do a single lab analysis to see what that snapshot reveals, to see if it shows connections to the data being found in the other sites around the basin, and to give us a clue as to whether it might be worth further developing those sites more in the future. 3 different funders now supporting this work, 1 private sector, 2 philanthropic. Because we now have these different sites all within one region, we're also working with everybody to start sort of what I'm going to call a regional study. It's not a scientific study per se, but it's while each of the sites has its own landowner or interest group and is working on the specific characteristics of that site. For example, one of them over here, let's say KLA 05 on the eastern shore of Upper Klamath Lake, KLA 03 is a 600-acre barley and potato farm, but nestled on the edge of the lake where they're developing restoring wetlands in between the agricultural land and the lakeshore in order to basically intercept the phosphorus that was flowing into the lake, contributing to the algae blooms, the toxicity of the lake, the deprivation of the oxygen, the destruction of both the historical fish habitat and waterfowl habitat. So this is a big restoration effort around reversing a lot of that. The site is called Lakeside Farms. We've been working there for 3 years. It's been their work has been very successful, independent of us. But what's been interesting is how the biomonitoring has been able to contribute specifically to their tracking of the presence of invasive plants that they're trying to remove, such as, for example, the yellow flag iris, and the restoration of native species, both ones which they are planting and trying to monitor. And so the data that we're providing is helping them track year to year the changes in the different species. But it's also helped identify a number of native plant species that have come back into the habitat without their planting. And so the assumption is that the increase in waterfowl that they're seeing, because of the restoration of the wetlands, the marshes along the lakeshore, which is bringing in some waterfowl in increased numbers that they weren't having before, they're assuming that the waterfowl are bringing in these other species with them that are taking root. And the bee data is showing that, and it's very exciting. Again, the bee data that we provide on the plant species breaks down into the concentration of the dominance of the different plants in the samples. So we're able to report, for example, what we would call a dominant species, which represents greater than 5% of the sample, but also trace species. Like maybe you've got, this X, Y, and Z are dominant, but look what showed up, a little bit of this, a little bit of that. And that's been really interesting at the Lakeside Farm site. At that one site, I'm just thinking through numbers to give you a real on-the-ground feeling. Over a summer season, we are now finding around 160 plant species per year. So you have a sense of the number of species that the bees are visiting and identifying across the landscape, which is more than anyone really expected there. More than half of those are of trace presence, a little bit of this, a little bit of that, maybe something a little farther away from the main site. We also are correlating those plant species as they roll up into the plant families that they correspond with the production of proteins or amino acids that are needed by pollinators. So we're able to report on the nutritional quality of the landscape itself for the bees. And in fact, that site is pretty healthy, but in some places we do this analysis, we might find that your landscape is lacking in tryptophan is one of the 10 aminos. And therefore, you might choose to cultivate in the following year some native species or that actually produce tryptophan to increase the quality of the habitat for the native species. Another thing going on at Lakeside Farms at that site is that, as I mentioned, they've got the invasive yellow flag iris. They're doing a lot of work to try to eradicate it. That included spraying of some pesticides, which the bees found, which surprised the farmers. They were like, "Oh my gosh, you caught us!" They were not using it at any level that was beyond the recommendations or the regulatory standards. Nevertheless, what we demonstrated with the bees is that pesticide was finding its way into other parts of the landscape. So the farmer has subsequently stopped spraying that pesticide, which is, I can't pronounce it, invasive, something like that is how it's pronounced. And they started applying it directly to the plants when they find them, as opposed to widespread spraying. We also found, I guess I'll just mention one more thing, interestingly, well, two things. We found gunpowder, which again surprised everybody. It surprised us. It took us a little while to figure out what was the source of this contaminant that we identified. And there's a lot of waterfowl hunting, and there's a skeet shooting range about 1.5 miles away. So the assumption is that some gunpowder drifted into the region or onto the plants that the bees are visiting. And we found some very interesting chemical residues 3 years ago, the first year we piloted, that seemed to correlate with the smoke from the forest fires. It was a very bad summer in that region, and some things showed up that nobody could quite get their head around in terms of contaminants in the bee pollen. And we made some loose approximations to identify that some of these could have been brought in the smoke from the fires. Final thing I wanted to mention in this region then is that because we now have this number of sites, the local stakeholders are interested in correlating their data with each other. So we are not at this point doing what I would call a scientific study, but we're scoping out with everybody what would we want to track across these 12 different sites. Specific plant species, specific ecological characteristics, trends over time. So I was just there 3 days ago. If you go to the bottom left of this map, you see 3 sites kind of close to each other. Those are clustered around one of the reservoir that has been drained as the J.C. Boyle. One of the big dams has been drained on the Klamath River. And so all the players involved, including the federal government, the tribal communities, the restoration company, and others, are now replanting the exposed riverbed with native plants. We are not monitoring that directly at this point, although we're in conversations with them about whether and how biomonitoring might directly support their effort to monitor the uptake of the plant species and the recovery or other changes that take place following the river restoration effort. But we do have these 3 discovery sites in the region, and it's going to be interesting just to see what we find. And I think that I hope it will include an indication of the uptake of the native plant species on the restored riverbed. It's also going to show a baseline of some of the upland species in the watershed. And there's a lot of curiosity around how might the upland habitat change in the next few years as the riverbed is restored, as hopefully the salmon begin to migrate upriver again. What's the overall impact? So I'm going to stop now, but I just wanted to throw those bunch of ideas out there to give you an indication of how we're implementing it, how the data is being used, and how dynamic it is, right? It's you end up with these data sets, and then we and you think about, okay, what does this mean? Where do we go with it? What do we want to analyze further? Do we want to dig back in and look for specific indicators? That sort of thing. Last thing in the US I would mention, I was at a conference in New Orleans recently, started a very interesting conversation with Ducks Unlimited. So we're having some fun discussions about how biomonitoring might play a role in some of the conservation work they get involved in. Up in Montana, we have a comparator site between approximately 7,000 acre regenerative organic grain farm and cattle ranch, and a conventional, not organic site nearby, a few miles away. Comparing and contrasting the plant species, the native species, the invasive species, and the correlations of those with the potential presence of different pesticides. And I would note one thing there, which is that the sensitivity of some of this analysis, we found during one early point in the testing, very high levels of lead concentration that was quite alarming. And then it went away, and the assumption is that some lead residue was blowing in from a mining region up in the hills, approximately 20 miles away. So a lot of this work in Europe too shows a real sensitivity to particles that are being aerosolized and drifting in the wind. Another thing we're going to do on the Upper Klamath Lake this year, we're going to test for cyanotoxins that are released by the algae blooms. We don't have any idea whether the bee pollen is going to pick these up or not, but we've documented and demonstrated in the laboratory that we can conduct the test. So we're going to run a few analyses this fall and see what we find. There's also one of those Klamath sites is on a former lumber mill site where we know that dioxins were used historically. In Europe, we have very successfully actively tested for dioxins in more active facilities like waste management facilities. So we're going to run some dioxins tests. It's a big experiment to figure out if and how we can, through the bee pollen, detect the presence of historical dioxin as well that's in proximity to a pretty sensitive river area. Cyril, why don't you go ahead, show the next slide. I want everybody to know that Tim here is on our team, but his schedule shifted this week, so he is on today on Shabiga Island off the coast of Maine, and he couldn't make this call. We have not done a lot of coastal marshland work, but Tim is our beekeeper in Maine, and that's what he does, a professional wetland scientist. So, and anything that we might discuss or be able to develop with any of you in the future, which we'd love to do, Tim would be a key piece of everything we do. I'm going to stop. Thank you for listening. Brittany, thanks for your patience. We'll just be quiet and hear feedback, questions, whatever would be helpful.
So we do have a question that they asked, how do you work with the local beekeepers? Awesome.
Yeah, so let me describe the implementation of a basic project, which is to say it's really pretty simple. All of our projects work with local beekeepers. So in every project, including all the Klamath ones I was just showing, it's the Klamath Basin Beekeepers Association who we partnered with. After your priorities are identified, like after we work with you, the local partner to determine what is it you're interested in and scoped out a project, we will work with you to identify a local beekeeper that can help manage the beehives and connect the pollen. We have to make sure their skills match some basic criteria. We provide about a 45-minute Zoom training just on the protocols for the sample collection and monitoring. We also ship the local beekeeper a small kit, which includes the attachment for their beehive, which is pretty standardized in terms of how they attach. It's pretty easy to attach to a beehive. that will help collect the daily pollen samples. We have a beekeeping team that is available, every day, all the time. The beekeeper sets up the hive at the site, attaches the device. We coach them through it if needed, but it's pretty straightforward. And then basically the beekeeper just does what they do anywhere else. It's just normal beekeeping, with an extra visit, usually every 3 weeks, to the beehive to collect the pollen samples that have been gathering in basically a small basket. So the bees are bringing pollen on a daily basis. Our methodology collects about 1% to 2% of the pollen from those bees without harming the bees. The beekeeper takes care of the bees as normal and collects those pollen samples and ships them to us. So for the beekeepers, from the beekeeper's point of view, it's pretty normal beekeeping in most cases, following a protocol that's important. And then they're usually, it depends on case by case, but they're usually pretty excited. It's interesting. It's fun. They learn new information about their bees and their beehives. They or you keep the honey and do what you want with the honey that the bees are producing. It's really this pollen sample that is then shipped to us on a regular basis.
I have one more question in the comments. It says, have you guys done work with the U.S. Agency for International Development, USAID, Bureau for Resilience, Environment, and Food Security?
No.
Again, no, we haven't. We're discussing with the Belgian equivalent of USAID for projects in Tanzania and Ivory Coast, for example. So Western and in Eastern Africa. We haven't been in contact with USAID, and if you have a contact, we'd be very happy to discuss with them. I do believe that for large restoration projects in Africa, we have a tool that is replicable in the US, but also in Africa or in other places. And so you can really compare the work on several ecosystems with a very easy, it's an easy way to do. And also very nice to communicate with local stakeholders.
All right. I have one more question in the chat, and it is, can you describe the process of setting up the hives? So I think you kind of answered that with the actual box, but
yeah, I mean, again, so I was in the Klamath River just a few days ago. We met with our local community partners. We went to the area. For example, I mentioned there's a former lumber mill site that's on the edge of a river. Walked the site with the beekeeper, the landowner and the beekeeper discussed where the beehives might go and came up with a good location that met all the qualities to keep the bees happy and also to make sure that the site is secure from either people in the neighborhood who might be concerned or particular wildlife. All these things are taken into consideration how you set up a hive. And in some places, we have a cattle ranch where we had to put a little barbed wire fence around the hive, a little bit of a radius to prevent the cattle from scratching their backs on the beehive. And so the site was identified. The 2 days later, we set up the beehives. Right, we had previously provided the beekeeper with the pollen collection attachment, which is the small patented device that we provide. The beehives were set up, and it's up and running. That's it. So the beekeeper sets up the hive as normal. They will go back. They will monitor pretty regularly for a handful of days to make sure everything looks good and the bees look happy. And after that, it's the visit for our purposes every 3 weeks to collect the pollen. In particular cases, again, we need to make sure we're working with skilled beekeepers. There may be additional care needed for the bees. if it's, especially because we want to capture the full year's pollen samples. And so we start early and we go late, and there may not be enough food and nutrition in the environment for the bees at the start of the end of the year, or if there's a particular drought condition or that sort of thing. So working with the beekeepers is important to understand anything that's going on in the environment, making sure that they're doing things like during the forest fires, the beekeeper out west, the beekeeper supplemented the bees with some common practice, basically sugar water, so that the bees would still forage the environment, but if they weren't getting enough calories, they had a supplemental food source. So it's important to be working with the beekeeper, again, in terms of what they do normally to care for the hive, which is important first for the bees, second for the data that we're collecting. I would repeat one of the scientific points, a piece of the foundation of what we're doing, which is that the bees are out foraging for amino acids for their diets, for aminos. They're going to cover, on average, about 1,800 acres over approximately 6 weeks. That's science, okay? They seek not just the nectar for the caloric intake, but for the proteins for the nutritional diversity they need. And so even in an environment where that is not rich typically for pollinators, if we install beehives, supplement the feeding if necessary to make sure the bees are not going hungry, they're still going to forage the entire zone, including a coastal marsh, right? They're going to look for whatever's out there and bring back small samples. So that's part of the benefit of working with these tens of thousands of daily sample collecting partners.
Yeah, I like that you call them employ bees. Employ bees.
Employ bees.
Yes, very cute. So yeah, one more question, and is wondering what costs are associated with the bee hotel installation, monitoring, data collection, reporting, and is that available in the U.S. now, or is that still just a pilot project for Europe?
So today it was designed for Western Europe, Megachilidae bees. So it will depend, I think, on the location depending on the state, because we need to make sure that the bees that might go in the bee hotel are normally or naturally present in the state or in the region where we want to do the test. So I can't answer for the whole states. It's huge. I know that it doesn't work for all places in Europe because of the specific taxes that we're looking for, or that the bee hotel is supposed to host. So I think it would depend, but it should work as such in most places. But we need to just do a quick cross-reference with the available data on what kind of natural or solitary bees or wild bees are present in the area and what kind of needs they have in terms of reproduction, refuge or shelter.
We have not yet done it in the U.S., but our prototyping is just wrapping up in Europe, and it's very successful. So we're eager to do it in the U.S., and it would sort of be a bit of an experiment the first time, right?
Okay. Are there any other questions? Anyone want to come off mute or enter any questions in the chat? All right.
Can I mention a couple more things, Brittany?
Yes, definitely.
Yeah, briefly, just a couple more ways that the data is being used in the U.S. now. One thing we didn't really emphasize is the power of this kind of information for local stakeholder convening and public interest in education is like super, super powerful. And I think it grabs people's attention. It's transparent, it's fun, it's objective. And so even in cases where there's some political tension, for example, I mentioned the farming and ranching up in Montana, there's some differences of opinion around, among some of the local players. But when you put a data set on the table that lists 50 or 60 or 70 plant species and data around them and numbers, it really tends to bring people together and in a constructive way. So that's something I would emphasize. That's really a rewarding piece of this work, is that people get excited, people want to know more. It's not controversial, and it's pretty fascinating. And there's almost always things being discovered that weren't anticipated locally. One of the ones in Montana, we found a plum tree in the data set, and everybody said there are no plum trees in the area. And we're like, there's a plum tree out there somewhere. And the beekeeper hunted around town, and we don't know that it was this plum tree, but he found a plum tree in someone's backyard a couple of miles away, right? So things like that happen that are sort of public interest stories that are very interesting. Also on the Scott River, which is a tributary of the Klamath, when we first started this there 3 years ago, near the town of Etna, California, the bees identified an endemic plant, an endemic species that had, according to the records, not been seen that far east previously. And so the local partners then went out and mobilized a little volunteer team and marched around the countryside till they actually found a small patch of this plant. So that was pretty exciting for local community and science and ecological interests as well. So it's pretty interesting what gets revealed is a part of the baseline data collection. And then for real impact, the ability to identify a priority, change your practices, and monitor within a year can be pretty powerful.